Hello everybody. Let me first wish you all a very happy Diwali. My name is Gwen Williams and I head up the international team at Universities Wales. So that's the body that represents all of the universities in Wales. I also manage the Global Wales project, and that is a partnership between the Welsh Government, the universities, the Higher Education Funding Council for Wales and the British Council in Wales. Now, India is a priority country for Global Wales, and indeed this morning we witnessed the signing of an MOU between the Welsh Government and the state government of Telangana in India. I think that's probably enough about me, though. The focus of this morning's Education Day session for Diwali is on collaborative research, and today we have the privilege of the company of Dr Anita Malhotra, a molecular geneticist and evolutionary biologist based at Bangor University. Now, Anita has worked all over Asia on elucidating the relationship of pit viper snakes that cause the majority of bites in many countries. The presentation that you're about to see gives us an insight into and into the partnerships that she has, and is an absolutely fascinating insight into this area. So I think if we could have the presentation now, that would be great.
Hello, I'm going to talk to you today about some of the work we've been doing at Bangor around solving the problem of snakebite in India. India is a tropical country and therefore has a very diverse snake fauna. Only about 60 out of 300 species are venomous, but of course when they come into close contact with humans, this can cause problems. Now, you may have heard that Australia has the most dangerous snakes in the world. And that may be true in terms of the amount of mice that they can kill when you inject their venom in a laboratory setting. But in actual fact, what determines how dangerous a snake is a function of several things. Firstly, how large it is, because the larger it is, the more venom it can inject, and Russell's viper gets to quite large sizes, between 1.5 meters. Secondly, how toxic its venom is, and again, Russell's viper is a very toxic snake. Toxicity is actually measured in a number of different ways depending on how you inject the mice with venom, whether it's directly into a vein or under the skin or into the body cavity and so on. And at least under some of these routes of administration, you can see that Russell's viper actually does have very toxic venom. But the most important thing really is how often it comes into contact with humans. And Australia it doesn't really compete because it has a very low population density, whereas India, of course, has one of the highest. And being a largely rurally distributed population, people regularly come into contact with the snake as they go about their daily activities. This is a diagram of a snakebite victim and the symptoms that snakebite might produce. As it's taken from Wikipedia, it's a nicely sanitized diagram. The victim looks calm and almost cheerful, which is certainly not the case in real life when snakebite happens. Now, the reality, of course, is very different from this rather sanitized diagram. And I'm just going to show you, to really drive home how severe this problem is, some of the actual pictures of snakebite victims. I won't keep them up very long because they are rather shocking. But you can see that some of these injuries, at least, are going to produce life-changing injuries in victims, even if they survive Russell's viper bite, that are going to have long-term consequences for them and their families. Now, it's not as if we don't know of how to treat a snake bite. The essential drugs, the cure for snake bite, that are commonly described as antivenoms, have been known for over 100 years. They were invented by Albert Calmette in what's now Vietnam in 1896 after a severe flooding event forced cobras into villages around what was then Saigon, where they bit large numbers of people. Essentially, it starts with obtaining the venom from the snake that you want to manufacture the antivenom against, whose bite you want to treat. This venom is then mixed with various substances that help to promote an immunogenic response in the host animal, and diluted as well, so that the animal doesn't suffer the toxic effects of a snake bite. It's injected in small boluses, usually below the skin. And the host animals are most typically horses, but they can be other larger mammals such as camels, sheep, even rabbits, and surprisingly perhaps chickens. And chickens are the exception in that the resulting antibodies that are made against these foreign proteins by the host animal are harvested not from the blood of the animal, but from its eggs. However, presently there is no commercial production of antivenom from chickens as far as I know. So more typically what would happen is that once the animal has manufactured antibodies over a course of several months, being continually inoculated with this with the venom mixture, the blood would then be harvested in controlled amounts, so not to damage the animal. And the antibodies are then harvested by usually simple centrifugation, which separates the solid parts of the blood, the platelets, the blood cells and so on, from the serum in which the antibodies are contained. The antivenom then can simply be the concentrated or dried serum from these animals. More typically, they would undergo some refinement, adjustment of pH, further processing of the antibodies, and they can also be lyophilized or dried so that the antivenom can be stored as a dry product and reconstituted when it's needed, which has the advantage of not requiring a cold chain to preserve the activity of the antivenom. India, in fact, has a well-established antivenom, which is made not just for a single species, but for 4 species of snake, which are thought to be the ones that cause the most bites throughout the country. And this is known as a polyvalent antivenom. So rather than using the venom of just a single species for the immunization of the host animals, you would use a combination of venoms from all these species. It's made by a number of different manufacturers, and it's available both in liquid and in dried formulations. So, given that there's a treatment, why do people still die from snakebite in India? And why do almost 60,000 people die every year in India from snakebite? Well, I said it was a complicated problem, and there are a number of factors which contribute to this. And I don't have time to talk about all of them, so I'll just give you a couple of case studies to illustrate some of these problems. The first case study concerns a 10-year-old girl called Shayla from Kerala, who was at school when she was bitten. And the teachers refused to believe Shayla when she said she'd been bitten by a snake. They kept her in school, and it wasn't until an hour later that her father arrived to take her to hospital. Unfortunately, as quite often happens, the first hospital she was taken to was either unable or unwilling to treat her, and she was referred on to another hospital. In fact, that hospital as well referred her onwards, and unfortunately she died on the way to the third hospital. The second case study is a friend of mine, a guy called Kaushik, who's a rescuer based in North Bengal. Now, snake rescuers occur all over India, and they are usually very dedicated individuals who remove snakes from conflict situations where they might bite people and release them in a different location. And they have to do this under a license from the forestry department because all snakes are protected in India. Now, Kaushik, despite being a very experienced snake rescuer, one day caught a very large black snake and didn't recognize it as a venomous snake because he had never seen a snake that large or of that color, which was venomous. He assumed it was a rat snake, which is a very common non-venomous snake, gets to large sizes and can be quite black in color. Unfortunately, it turned out to be a species of fairly unusual krait, which is a highly venomous snake that can reach much larger sizes than the common krait, which is, as the name suggests, the one that's commonly encountered. Later on, they realized that a mistake had been made and it was in fact a krait, and he was rushed to hospital. Of course, the antivenom wasn't applicable. It didn't apply to this species of krait, Wall's krait, it turned out to be, and he went into a coma. He was in a coma for 3 days. Artificial ventilation was a lifesaver for him because a krait has very neurotoxic venom which paralyzes the respiratory muscles. And he was actually very lucky that he was able to access an available machine at a private hospital, because quite often these are pre-booked for all sorts of other issues and are not available to snakebite victims. As the title suggests, snakebite is a complex problem and has to be tackled at multiple levels to be effective. And each of these levels involves multiple stakeholders. And we also need to tackle them at various timescales. First of all, we have the short-term fixes, which is basically improving what we do already, but doing it better. And then there are the longer-term changes, the game changers, that can revolutionize the treatment of snakebite. At Bangor, we've mostly been involved in the basic research. We've also been involved in raising awareness and snakebite education in the communities that we work in. So this map that is on this slide is a depiction of the death rate across India, and it comes from a study called the Million Death Study, which investigated various causes of death among the population of India, not just snake bites, over a period starting in 2001. And you can see that the death rate can exceed 30 people per 100,000 head of population. And that's really quite an incredible death rate. It's about 3%. So the sharp-eyed people among you might have noticed in the previous slide where there were insets showing the range of the big four species, that there were blank areas on that map. And in fact, it turns out that some parts of India don't have any of the big four species at all, and instead they're replaced by a whole set of different species. And this particularly applies to two regions, the Himalayan regions and the Northeast Indian states. And this is because they belong to entirely different biogeographical zones to the rest of India. So to take as example, the spectacled cobra, a very iconic species. And this is one of the big four for which antivenom is available. But in these two regions, they are absent, or at least only partly present, and are replaced by other species of cobras. So in the northeastern states, we have the monocled cobra, Najacaeuthia, which actually extends quite far west in Bihar and Uttar Pradesh, and coexists with the spectacled cobra in West Bengal. And in the northwestern part of India, we have the Caspian or Central Asian cobra, which occurs at higher elevations than the spectacled cobra. And these two species are not presently covered by the antivenom. But even if we look just at the single species of cobra that is found over most of India and for which antivenom is available, Naja the spectacled cobra. we can actually see that there's a lot of venom variation within this species as well. So in the inset here, we see some experiments that were done on the effect of cobra venom on muscle. This is a section of the gastrocnemius muscle, basically the calf muscle of a rat. In the upper left corner is the control. Compare this to muscle that has been incubated with cobra venom from different parts of India. The purple dots here indicate inflammatory cells. So you can see that the cobra venom from West Bengal has created massive inflammation, which is not found in the other regions. Whereas the one from Mumbai, you can see all these white gaps, which basically indicate that the muscle has been digested away. In other words, it shows massive necrosis of the muscle. So it's very likely that the venom from Tamil Nadu, which is where the venom that is used for manufacture of antivenom is from, which shows neither massive inflammation or high levels of necrosis, lacks the toxins that produce these effects, and hence the antivenom may lack the antibodies which will counteract these effects and prevent them in snakebite victims from these other regions. So one of the states we focused on is the state of Himachal Pradesh, up here in the north of the country. It's a very diverse state. Most of the state lies in the foothills of the Himalayas, very mountainous, ranging right up to 6,800 meters. And at these highest elevations, you've got really cold deserts. But the lower elevations of the state, along the borders of Haryana and Punjab, for example, are much lower, about 300 meters, and are perfect conditions for venomous snakes and contain at least 3 of the 4 big 4 species. But there's very little information from Himachal Pradesh about snakes and snakebite. These are the venomous species that we've recorded in Himachal Pradesh so far. The ones outlined in red are the big 4 species. Only the saw-scaled viper has not yet been recorded in Himachal Pradesh, even at lower elevations. So you might think, and it's often stated that snakebite is less of a problem at higher elevations. But in fact, there are several venomous species that are quite common at higher elevations. And worthy of note here is the Himalayan pit viper, Gloides himalayanus, which in fact holds the record for the highest occurring snake ever found. It was recorded at just under 5,000 meters at the foot of a glacier in Himachal Pradesh in the early part of last century. Many of the species are only found at relatively low elevations, such as the black morph of the spectacled cobra and the common crate. Other species do extend to quite high elevations. Even Russell's viper, which was formerly thought to only occur below about 1,000 or 1,200 meters, we have actually found in Himachal Pradesh at over 2,000 meters in areas that would definitely be covered in snow during the winter. Another high elevation species is that other species of cobra that I mentioned earlier, the Central Asian cobra. And these 2 cobra species seem to be separated along an altitudinal gradient. It's also worth highlighting the green pit viper that is found in this state, which is commonly referred to as the northern white-lipped pit viper, Trimeresurus septentrionalis. Now, there's a common belief that green pit vipers have fairly mild toxicity and are not really worth bothering with when it comes to antivenom. However, we have had reports from physicians treating snakebite in some of the mid-elevation districts where these snakes are very common and cause more than 70% of the bites seen in those districts, that quite severe can't say this. They cause quite severe clotting disorders of the blood. In one report, an 8-year-old boy who'd been bitten by one of these species still had incoagulable blood after 8 days. Another state we've more recently been working on is Sikkim, which is a small state that's inserted between Nepal to the east and China to the west. Again, having this huge altitudinal gradient, but even more exaggerated, with about 250 meters along the border of West Bengal, right up to 8,000 meters here in West Sikkim, where Kanchenjunga, the third highest mountain in the world, is located. These are some of the species that we've found in Sikkim, the venomous species, including this Protobothrops himalayanus. All these names are unpronounceable. And this species was actually only described a few years ago, although it is a very large species, well over a meter in length, and you'd think it would be difficult to overlook it. On the bottom right-hand corner, we have the mountain pit viper, which is one of the commonest species and probably causes the most bites in this particular state and in other northeastern states as well. And at the top here, we have a really beautiful snake, which is again one of the lesser known species of krait, highly neurotoxic snake, which occupies the northeastern region of India. And in this particular one, it's again a relatively high elevation species. None of these species are covered by the current antivenom. So a brief description of what we actually do on our surveys. We basically get together a large team of people. These are drawn from our Indian collaborators as well as from the UK. We put a lot of time into walking up hillsides, searching both during the day and at night on foot, but also covering large distances by car where roads travel through suitable habitat. Once we've obtained our snakes, we temporarily keep them, return to our field base so that we can process them, and this involves taking some morphological data in order to confirm our identifications. We then sample the venom by allowing the snake to bite through a membrane that covers the receptacle. This venom is then dried and sent off for biochemical analysis by some of our collaborators in India. We then take a small sample of blood from the tail of the snake, which allows us later to extract DNA from it and to obtain sequences for us, again, to confirm identity through genetic means, understand the relationships of the snake, and perhaps be able to evaluate whether there are any cryptic species within what we currently understand to be a single species. And again, all this work is done in India. So to move to some of the results that we've obtained from this work, we put this data together and construct a phylogenetic tree. And this is just a small section of the phylogenetic tree that we've produced, showing just the vipers. And the upper part here are the pit vipers, and down in this lower section here, we have the true vipers. In India, these are just the saw-scale viper and Russell's viper. And taking a closer look at these individual clades, we can see a pattern that appears again and again across various snake species in India. And that is of a distinct genetic break between southern populations and northern populations. But then that does raise the question whether some of the variation that we see in different Russell viper populations and saw-scale viper populations is actually attributable to this cryptic genetic diversity that isn't currently reflected in the taxonomy at all in the case of Russell's viper, and in Equus is reflected in describing these two populations as different subspecies. Here again, in the case of the common crate, Bungus ceruleus, we see the same north-south split, although we have much less data here from the south at present. The exception to this pattern that I've described of a north-south split, strangely enough, it's not seen in the spectacle cobra at all, despite the fact that it is distributed right from the southern tip of India right up into the northern states such as Himachal Pradesh. There's very, very little genetic structure seen right across its range. I've highlighted here in this green box the samples that came from Himachal Pradesh, this black morph, just to reiterate that it isn't a different species, even though it looks very different, but is in fact genetically identical to more typical morph that occurs in other parts of India. Moving on to venom. The venom is analyzed using a method known as reverse phase high-performance liquid chromatography. And this is done by collaborators in Tezpur University who specialize in the analysis of venoms. And basically, this method relies on different toxins having different solubility in different solvents. And it uses a combination of two solvents, which range from 0 to 100% in concentration across a gradient. And as the optimum solubility is reached, these toxins are drawn into the column and are detected. So each of these peaks may have one or more toxins of similar solubility contained within them. But basically, they give us a fingerprint with which we can compare venom compositions between species and between populations of the same species. And here we're looking at the venom from 3 different populations of the spectacle cobra. And you can see that the venom composition is very similar with peaks in similar places. Some of these peaks differ in height, which indicates that these toxins are much more prevalent in the venoms of some populations than in others. It could mean that the antibodies that are generated against the toxins contained in these 2 peaks in the Tamil Nadu venom are not sufficient to fully neutralize the toxins in these peaks in the venom of the Himachal Pradesh cobra, and could account for the increased severity of bites and the inability of the antivenom to treat cobra bites in Himachal Pradesh. We can also more directly measure the neutralization of the venoms with the existing antivenom by actually incubating the venom together with the antivenom for a period of time, and then seeing what hasn't been bound by the antivenom, which is immobilized on some kind of matrix. So we see the results of these experiments here in the case of the spectacle cobra from Tamil Nadu. This, of course, is the species and the population for which the antivenom is actually manufactured. And we can see that only very small traces of the toxin peaks remain after treatment with the antivenom. On the other hand, when we treat the venom of a different species, the Central Caspian cobra, Central Asian cobra, in a similar way, we can see, perhaps unsurprisingly, that there are much larger amounts of toxins left over after neutralization with the same amount of antivenom. So finally, just returning to the bites of the green pit viper in Himachal Pradesh, where I mentioned we saw blood coagulation disorder persisting for some days. Now, when I say blood coagulation disorder, I mean that the blood doesn't clot and continues to bleed and can cause severe blood loss in bites in victims of this species. However, this slide mentions procoagulant activity, and as you can see here, it actually decreases the clotting time. So how does this sort of, how do these 2 facts coexist? Well, in fact, the procoagulant activity of some snake venoms is so strong that it actually uses up all the clotting factors in the blood. It can take days for the body to replenish these factors, and if the toxins are still circulating in the blood, then they can be used up just as quickly as they are replenished. Now, I also mentioned that green pit viper venoms are often thought to be much milder and less toxic than things like Russell's viper. But note here that we've actually used Russell's viper venom as a control, and although here in relation to the normal clotting time, the NCT, there's a dose-dependent reaction, so that at lower dosages, there's not much difference from the normal clotting time, but then it drastically drops, and at similar concentrations to Russell's viper venom has almost the same procoagulant activity. One of the quickest and maybe cheapest ways of reducing the death toll from snakebite is to actually prevent snake bites happening in the first place. And so, alongside the work that we've been doing related to basic research, we've also been conducting education and awareness workshops for communities and especially for schools. At these events, we distribute posters that we've made that depict ways in which snakebite can be prevented, and also stickers and posters that depict the difference between venomous and non-venomous snakes for the region in question, and these are all translated into local languages. The Forest Department staff really play a key role in the resolution of human-wildlife conflict in India, but generally they consider wildlife to be the more charismatic, the larger things like leopards, elephants and bears, and of course they do cause conflict in many parts of India. It's a very important part of their remit, but snakes probably cause far more instances of conflict and far more deaths than any of those big charismatic mammals. So in parts of the country, again, where there aren't many rescuers from outside the Forest Department who they can use, we've started training the forest officials themselves. We've conducted training events for about 200 different forest officials in Himachal Pradesh, about 100 in Sikkim. And as well as training them how to handle, we provide the equipment with which to do so safely, the equipment that we use ourselves. It's not easily available in India, but we've worked out how to manufacture it there, but it's still fairly expensive. So Bangor has helped to provide funds to manufacture these, to hand out to forestry departments. We have workshops where we provide them some basic information about snakes, their role in the ecosystem, their biology, and help them to include snake conflict in their human-wildlife conflict activities. At a different level, we've also held workshops aimed at providing advice to policymakers on how we can address the various issues relating to snakebite. And this was held in December 2017 in Hyderabad. And in the foreground here, you've got the doctors, the clinicians who are actually involved and have a lot of experience at treating snakebite from all across South Asia, discussing their area together. And in the background, we have various other groups discussing education and awareness, basic research, and so on. And we put together, as a consequence of this workshop, a policy brief, which can be downloaded from our program website. So thank you very much for listening, and I would just like to highlight that all this work has been collaborative. It's absolutely essential in India, and we collaborate with a wide variety of universities, other research institutions, not-for-profit organizations, and the forest departments themselves. And of course, the funding is very important, and we have had major funding from the EU, as well as from the Economic and Social Research Council through the Bangor Fund, and the Rufford organisation has also funded some of the field work.
Gosh, what an absolutely fascinating topic that is. I've got the privilege of having Dr. Anita Malhotra with us here now to answer any of your questions. So could I please encourage the audience to submit and might have through the Q&A, and I will field them to Anita. In the meantime, Anita, it really begs the question, how did you even start get to work with venomous snakes? Tell us about your story.
Well, I was born and I grew up in India, and I've always been interested in animals. I mean, you put these fancy labels on me, evolutionary biologist and so on. Those come later. In the beginning, it's just, you have a fascination with animals. And eventually this focused on being fascinated with reptiles and in particular snakes. And as I went through my career, this began, directing what I was working on. So the early part of my career was very much focused on studying pit vipers, as you mentioned, and very much all over Asia. They really were a mess. No one could tell them apart. No one knew where the species lived. So I spent perhaps 2 decades working on that. So I was very much focused on the animals at first, and not really considering people. But, as you carry on working on venomous snakes, it becomes impossible to actually ignore the fact that they do have a very negative effect on a lot of people. And it just began to make me feel that I had to do something about it. I couldn't really justify just focusing on the animals without, looking at the human snake interface and helping people as well as snakes, because, of course, the more negative attitudes people have towards snakes, the more the snakes suffer as they persecuted.
Yeah.
So it's both sides of it now for me.
Yeah. And a particularly tragic story as part of your presentation there as well. I noted that. Next question from me, I suppose, while I wait for some to come in on the chat, is, I'm conscious that India is vast, and biologically very, very diverse. I mean, is it possible to design an anti-venom that will work right across the country, or is there an alternative solution to that?
Well, the problem with the antivenom in the way that it's manufactured at the moment is that because the whole venom is injected into horses or whatever to manufacture the antibodies, essentially they make antibodies against anything that's in the venom, even if it's not actually what's killing people or causing damage to people. And there is, the more venoms you inject, the more of the variety of these proteins and other toxins, and you get lower antibody as being prepared against any particular toxin. So the more snake venoms you include, the less effective it becomes against the toxins of any one particular species. So there is a limit really there in how many snakes you can cover using conventional methods. But there are lots and lots of different research programs at the moment trying to find other ways of doing it, using more modern technology and a more detailed understanding of the venom and the toxins that are contained in it, to perhaps either produce it in artificial circumstances, in a lab entirely, or by actually producing or selecting more carefully the toxins that we inject into the horses in order to just select antibodies against the most important, the most lethal and damaging toxins. So there is some hope, but of course all these things are still in the research phase, and actually translating those into something that's available for treatment is a long road. And I think coronavirus has brought that very much to everybody's attention, that it can take a very long time to translate even very promising research into something that's actually available for treatment. But the other way to do it perhaps is to, rather than trying to cover the whole of India in a single antivenom, is to have more regional preparations that are suitable, perhaps, say, for the south, for the northeast especially.
Yes.
Since it's really different from a herpetological point of view to the rest of India. But still, we need the basic knowledge in order to decide, where the the boundaries lie between these different regions, and which are the snakes that are important in these different regions. Because even in just northeast India, there's still a vast number of species that could be covered. There's 6 species of kraits, for example, in that area alone. Do we try and cover all of them, or do we try and find out which are the ones that are the most significant when it comes to bites? So there's still a lot of basic information that's needed, and that's really where I feel my role is actually providing some of that baseline information.
That's fascinating. Thank you. The questions are really beginning to come in now on the Q&A. You were talking about the production of antivenom, and some of them relate to that. And somebody was asking how is the antivenom mass produced? Does it come from the snakes, or can it be synthesized?
Well, it does at the moment. It's come from you start from the snake venom, and goes through the process of being injected into horses. So obviously, to mass produce it, you need quite a lot of horses.
Yeah.
It's not a cheap thing to do, because of course the horses have to be very carefully looked after. They have to be healthy. They have to be, because you are injecting toxins into them. They need long-term veterinary care, and lots of land in which to run these large herds of horses.
Yes, and this leads on very nicely to another question, actually, which is, when the antivenom is being created in an animal like a horse, can it be harmful to the animal, and what are the ethical considerations there? I mean, you've already touched on this to an extent.
Oh, I mean, there certainly are ethical considerations, and it does have to be done very carefully and under good veterinary supervision. I mean, the aim is not to, horses are valuable animals. The aim of the manufacturers is to keep their animals healthy. So, the immunization protocols are themselves the subject of extensive research. So it does have to be done in a very regulated way.
Okay, thank you. Just going on to another question that we've received through the chat, through the Q&A. Have there been any species that have been particularly difficult to work with, and if so, why?
Yes, from different points of view. Some animals are just difficult to find. Even if they do bite a lot of people, actually, they are very cryptic animals. They are nocturnal. They hide away quite effectively at night. Some animals are difficult to work with in the sense that they're very difficult to obtain venom from. So vipers are generally quite ready to bite. They have long fangs and large venom glands, and they produce lots of it. When you're working with some of the smaller species, of course, you're obtaining very small amounts of venom, like for the saw-scale vipers. And kraits have actually quite small fangs, and you can't milk them the conventional way. Getting them, as I showed, getting them to bite through a membrane. We use different processes for that. We actually use a pipette tip, and to milk them over each fang and milk them directly into the pipette tips. Otherwise, you lose too much of the venom, and even with quite large snakes in quite small amounts. So actually obtaining enough venom for research can be a challenge with those sorts of species.
How interesting. The other question that's come in, do you raise awareness about the taipan?
No, it's obviously an Australian species, and that's not somewhere that I have worked. So yeah, I haven't actually. We focus on snakebite awareness in India on Indian species.
I'm glad you clarified that one for me. I wasn't sure what a taipan was there. Another question from me, I suppose. Then, in rural parts of India, what do people do if they're bitten and they find themselves far away from hospital?
Well, that's a very good question. That's one of the remaining areas that is very challenging, is how to, first of all, actually make people aware that they do need to go to hospital. It's actually surprising that in this day and age, there are a lot of people who have never heard of antivenom and don't know that they can go and get a life-saving treatment if they go to hospital. So that's one of the first things. And actually, it's quite nice that some of the work in Himachal Pradesh that we've been doing and explaining to people like the disaster management committees, they've actually started putting out a radio jingle now that tells people what to do, go to hospital immediately. And also in some states, there are now free ambulance services.
Yeah.
So there is there's a toll-free number that you can dial and summon an ambulance. And the number varies from state to state, but it's frequently 108 if anyone's listening in India. And it's a very, very modern ambulance fleet. It's all computer controlled. And they have a very, very good record of reaching people even in quite remote areas, as you can imagine in Himachal Pradesh, where you're often dealing with very narrow mountain roads that are frequently closed by landslides in the wet season. It can be a real challenge, but as well as four-wheel drive ambulances, they also have motorbikes, boats in some parts of India where communication is largely by the rivers. And they do reach a large majority of people and in sufficient time to get them to hospital to get treatment. I mean, that's perhaps the best case scenario, and maybe it doesn't work just quite as well in all parts of India. But that is definitely something that's available and can be worked on and can be improved in areas where it's not working so well.
Okay,
So in theory, it's possible to get people to hospital within the so-called golden hour,
yes.
to get them treated.
Yeah, the challenges are enormous, clearly, but yes, that's good. That's good to know. Thank you very much, Anita. I mean, I'm conscious of the time. I think we've just drawing to an end, just as the clock strikes 12. So all that remains for me to say really is a huge thank you for taking part. It's been absolutely fascinating to get an insight into your work and the partnership that happens between universities here in Wales and universities in India as well. And thank you also. To the audience for listening in and for submitting their questions. And I think all that remains is for me to wish everybody a very happy Diwali.